So guys, we can start with Dr. Sen. Just a little bit about what you do so audience knows kind of who best to address their questions towards.
Sure. So I am a biochemist by training, spent about 25 years in biotech and pharmaceutical industry. About 12 years ago, got into plant chemicals, if you will. I had worked with synthetic delta-9 THC back in 1977. Came back about 3 years ago, looking at the whole plant. So what we are focusing on is presenting the whole plant portfolio of phytochemicals, both in water solutions as well as in free oils.
Okay.
Dr. Sen.
And I'm Jennifer Stamps. I'm a clinical neuroscientist for trait. My background, my basic science background is in human chemosensory function, taste and smell, flavor perception. And then my medical background is in neurology. And I came from academia in the clinic and joined trait back in September. We have a multi-platform for water solubility, true water solubility. That's we have all our methods are natural. It can be done in yeast, plant, non-GMO, GMO. Anyway, we can talk more about that, I guess, during the panel. And
Alexey Peshkovsky. I am President and Chief Scientific Officer of Industrial Sound Mechanics. We've been around for about 14 years. We were formed with the pharmaceutical industry in mind. Our model is to enable other companies to make their own water-compatible bioactive formulations, hydrophobic bioactive formulations. So for the pharmaceutical industry, this is what we basically designed all the technology around, the ability to scale up. We use ultrasonic technology. It's called barbell horn ultrasonic technology that allows one to scale up from lab to industrial level without losing the intensity or changing the quality of the final product. And we added some other capabilities, nano stabilizer, specifically it's the rest of the formulation, so the other piece of the puzzle. We worked with the pharma industry for the first about 10 years, but then the cannabis industry just exploded with the same need to deliver a hydrophobic bioactive, which is THC, CBD, cannabis extract oils, into the bloodstream. So we basically got lucky. We had everything designed, and then the cannabis industry created a lot of demand. So this is what we do.
Also, I have some questions if we're feeling shy and we don't want to start with audience questions, but if we do have audience questions, I'm happy to start out there as well. I want to make sure everyone has time. Do I have Daniel? Are you here with the mic? No? Okay.
So my question is about nano emulsion. I know that you guys are all working on some pretty interesting new ways of getting cannabis into a stable state in water, which is awesome. We in the industry have heard a lot of concerns about the safety of nano emulsion and the potential for it to build up in the brain, and I was wondering if you could comment on that.
I go? Okay. So nanotechnology and nanoemulsions and all these nano terms are relatively new, but nanoemulsions are not. I'll have a presentation later. I'll show some particle size distribution. Tea, coffee, beer, milk, lots of other things are all nanoemulsions. They just weren't called that, but they've been around for a while. In order to bring hydrophobic substances into your bloodstream, you create something called mixed micelle suspension in the small intestine, which is also a type of nanoemulsion. So nanotechnology could be dangerous, of course, and it mostly relates to solid and biopersistent nanoparticles, things like carbon nanotubes and metal oxides. But if all the ingredients are food grade, if they are all present in the human body anyway, or close to it, then it's likely that it's safe. There's also sometimes a misconception. Many people believe that the entire nanoparticle gets absorbed, and that's, I shouldn't say 100%, but it's mostly not so. the nanoparticle is necessary in order to deliver the cannabinoids to the absorption site, to the enterocyte cells. After that, it falls apart, and molecules go in as they always do.
Also, at Trait, we make truly water-soluble cannabinoids that are naturally, and to avoid nano-emulsions for a few reasons. One, we're in negotiations with a lot of major flavor and ingredient formulation companies, and the big companies are staying away from nano-emulsions due to the risks that are unknown and possible liability later. Also, the tendency for things to bioaccumulate from nano-emulsions are known and unknown. So there's some problems with that. You know, you can get pesticides from your plant that bioaccumulate in the brain because of nano-emulsions. So it's not just the cannabinoids that are being nano-emulsed. You know, it's the pesticides and fungicides as well. So, and also the taste. You have that soapy taste. So our platforms are truly water soluble to avoid all of that. Yeah.
I think a couple of quick comments about sort of the nanotechnology, so to speak. It's been around for over 50 years. So unfortunately, in this industry, because of lack of good science, it's bundled into this itty-bitty tiny science. It's not. And as Alex and Jennifer pointed out, there are multiple ways of creating nanoparticle, nanoemulsion, nano what have you. It's not a single platform technology. Depending on how it's done, the effect and consequence can be all over the map. In fact, one of the most successful yet unanticipated story with nanoemulsion happened back in the mid-'80s. An Argus Pharmaceutical, a small biotech company in Houston, made nanoparticles with AZT, the first HIV drug, and it worked like a miracle. And they thought, oh, we've finally solved drug delivery, this is how everything should be done. Only to find out 6 years later that in that format, the drug accumulated in liver, specifically a group of cells called phagocytes, which is also where most of the HIV virus gets sequestered. So no wonder it worked wonderfully. You literally put the drug targeted to what the virus is. So, you know, the concern about any kind of chemical formulation technology is that, number one, we cannot predict exactly what's going to happen. It takes years, if not decades, to figure it out, and it depends on what it is that you're formulating. But, you know, and that's genetically true for any technology. You know, from natural to quote-unquote truly water-soluble, it can have very, very interesting consequences. And, you know, many of you probably do remember after plastic bottled water first came out, it took 10 years before BPA was addressed, right? So a lot of things kind of putting together very generic answer and ask very standard questions. In a human system, it will never work out that way. So it's going to be variable, and that's why the monitoring is really, really important. Why are you doing it? What's the purpose? If it's solely for taste, as was pointed out, then you can probably keep or create an appropriate method to reduce the taste issue, but then you don't know what the other consequence is going to be. Would it accumulate in a different organ that you don't want it to go to, right? So oversimplifying has been a real issue in this industry. And, you know, I've got a presentation later on, and that has been my biggest beef coming into this field 40 years later, after I first worked with Delta-9 THC as a pure chemically synthesized lipid, to come back and find people that still putz around with oils.
We have a question here, and then one over there, and one here. So I'm keeping track of order. You may start.
Hi. I had a question about biosynthesis and whether there's a difference in terms of beverage technology if you're using a synthetic cannabinoid versus a plant-derived one.
I think you need to probably separate out biosynthesis versus chemical synthesis. The origin of biotech industry was finding ways to biosynthesize chemical intermediates. So that is done by the synthetic machinery of a living cell, which relies on enzymes. You can do the same thing on a bench top, either chemically or having the enzyme in a little test tube carrying out the same reaction. Okay, so biosynthesis is more like what is being done at Trait, what's being done at Demetrix, who is cloning all the genes in a yeast and trying to produce the finished cannabinoids. Chemical synthesis is way more defined. You know what kinds of chemical components and impurities are likely to be there. That is the foundation of the pharmaceutical industry. So they could very easily figure out what to look for in terms of adverse events. When we started biotech industry back in the 70s, we actually had to work with the FDA to discuss what are the concerns. None of us knew, right? And that was the origin of what's called points to consider. It was an industry FDA collaboration to say, what should we fear? And if you make insulin in a bacteria that produces endotoxin, how are you going to deal with that, right? So the issue about biosynthesis is obviously it mimics more the natural path. If Mother Nature could make it, this is how she would do it. The problem is she does a whole bunch of other things that we don't know what they are.
Hi, I have a question. Over the last several months, I've spoken to a number of producers, processors making this isolate, and every time I talk to somebody, one person says there's no such thing as water-soluble CBD and THC, and then someone says they have a water-soluble THC. And so, I'm not a scientist, but trying to figure out why is there that disconnect? Is there different spectrums of soluble, or is it just some people figure it out and some people haven't?
Again, yeah, go ahead. So, by definition, every single molecule known to man will dissolve in something. Some will dissolve naturally in alcohol, others in water, others in oil. Some will dissolve in something, not in others. Then there is different extent of solubility. If you go into your kitchen, pick up table salt and baking soda. They're both sodium salts. One dissolves very well, table salt, in water. Baking soda doesn't. So if you're only looking at the sodium part of it, you will say, gee, who is right? Are sodium salts soluble or not? So lipids, by definition, what all the natural molecules, CBD, THC, in your body, cholesterol, in plants, chlorophyll, the green pigment, those are all in their pure form, lipids. They are in that state, never water soluble, never. However, you can do all kinds of things, as Alexey pointed out, to make them water compatible, or chemically modify them. That can be done either in a test tube or in a GMO plant that will alter the molecule to make them water soluble, but it's a different molecule.
Also, there are natural ways to make the cannabinoids water soluble. So we glycosylate the cannabinoids. We can do that in the plant, which will also increase yield by 4 to 10x. And I'll show you, this is a nano emulsion. You can see, this is a CBD nano emulsion. This is our water-soluble CBD. It tastes like water, it's odorless, it feels like water. And then here is CBD just in water, not nano-emulsed, and it's very separated, So we have 5 different natural ways to make cannabinoids and other plant compounds water-soluble, truly water-soluble and naturally. We can do it in the plant, like I said. We can scale up with yeast and in vivo applications also that are highly scalable. So we're doing it. This is at Trait. We have several ways. There's a protein binding method that I came up with from the olfactory system. I can't tell you a lot about it, but I can tell you that that's one of the methods we use, and that's completely natural. And it's what the body does naturally anyway to make a compound hydrophilic that was hydrophobic so that it can interact with the receptor. And our glycosylated pathway, that platform, I can talk more about that because that patent's been published, or those patents are public now. So we just attach sugar molecules onto the cannabinoid, and it's completely water soluble. It looks like this, and it tastes like water. So there's no soapy taste or bitterness from the cannabinoid. If you have any, yeah.
Let me address that as well. So we get that question a lot too. I think it's just about the terminology. If you're trained as a chemist, then you take the word solubility seriously. Otherwise, you might not take it so seriously. So when it goes into water and it looks like it mixed in very well and you don't see it anymore, or maybe it's homogeneous and it's not going to separate, people just decide, okay, let's call that water soluble. I think most of those things are not water soluble. I like to call them water compatible, unless, right, exactly. So
Ours is.
Yeah, the difference is
Right.
Water compatible.
So
It's actually
chemically
water
altered.
So if we're going to stay with the word soluble,
or protein bound.
soluble means it's able to separate on its own into molecules like salt or sugar does in water and kind of disappear into it. Water compatible, which is the term I prefer, is for all intents and purposes it looks like it's dissolved, but if you really look into it, then there may be some small droplets that you don't see that don't interfere with light, so everything is transparent. But very technically speaking, it's not dissolved, but for all intents and purposes, it acts as if it were. Cannabinoids are just simply not soluble in water. They're just not, unless you modify them. So if you do what they do, you can get them to dissolve, but for that they have to be synthetic. They could be done in a very clever and possibly safe way. I'm not exactly sure what the regulations are on that.
It's a natural process. And
Okay.
It goes back to the parent compound after the oral phase.
Yeah. So if you modify them, they're no longer the original cannabinoids, but they might be close, and you can make them truly soluble that way. Otherwise, you could just nano-emulsify them using also natural and safe ingredients, and then they will behave as if they were water soluble, but very technically won't be.
And they separate. The shelf stability isn't as good either.
That's
That's a problem. Yeah.
true.
Oh.
They don't separate at all. They stay for years and years. Oh, is that me? Am I exploding?
Don't move.
With which part of my body am I doing this?
Wherever the transponder is, I think.
Yeah.
Yeah.
What is the shelf stability, and from a consumer standpoint, you're not going to put it under a microscope. What is the distinction matter, consumer to beverage?
You can get the salad dressing effect where all the cannabinoids are on the layer at the very top, and your first sip is just an oily cannabinoid sip, and the rest of the beverage is.
They shake it or you homogenize it, does that solve that? Or
We talk about nano-emulsions or
To some degree,
or
but yeah, it eventually they separate out.
I think one thing that you need to realize, there is a huge difference between water and multi-component beverage. 5-Hour Energy is a beverage. Nestle Pure Life is just water. So when you ask about shelf stability, any molecule will be different in different matrices. So from consumer perspective, it would be more, is there an off taste? Is it no longer delivering the cognitive effect? Those are much better indicators than measuring THC.
post-production stability methods like HPP or Retor or something like that, do they work?
Are you asking about nano-emulsions or the modified soluble cannabinoids?
I think either or both. Sure.
I can address the nanoemulsion bit.
And I can do the water-soluble bit.
Okay, let's do it. So nanoemulsions are interesting. Oh, am I? Am I being demoted or promoted?
There you go.
There you go, yeah.
It's on, right? Okay. Oh, so much simpler.
Don't move.
Definitely not nanotechnology. Nanoemulsions are in a stability pocket. They are kinetically stable permanently if you make them right. So it depends on what the droplet size is and what the surfactants are. So if your formulation is correctly prepared and if you have the right equipment that takes you down to about 300 nanometers or below for the entire distribution, preferably below 100 nanometers, those will never separate because Brownian motion by far supersedes any systematic Archimedes force-based phenomenon. Larger droplets float because of the density difference, and it's not sufficiently a self-stirring system when the droplets are large because Brownian motion is not large enough. When you get down to below 300 nanometers and most certainly below 100 nanometers, just random motion supersedes by far any systematic motion, so it just stays in suspension. We have some nanoemulsions we made years ago. I think for the pharma we made some well over 5 years ago, and they're completely unchanged. We also monitor the potency with HPLC, and there is a way to lose potency if you expose it to light or something like that, but that would be on the oxidation side, not because of how it's formulated. It's just sometimes cannabinoids can get destroyed by external factors or by a fungus eating it if you don't sterilize it or something like that. Otherwise, they're permanently stable.
Same for the water-soluble. They're very stable, especially the protein-bound platform. It's permanently stable as well. The big difference is the taste. There isn't one, so that's really helpful. Yeah.
A comment.
And there's no salad dressing effect. You know, it's just water.
A comment that needs to be added to that is that when Mother Nature wants to make a lipid water soluble, she has three different ways. One is chemical modification like glycosylation, phosphorylation, and the like. And that happens to all natural molecules in a human system versus yeast. It doesn't happen in bacteria because it's a very lower life form. The other, which is quite common, and if you go get your blood drawn now, as opposed to back in the 70s when I could get my blood drawn, back then they would measure total cholesterol, total triglyceride. So they'll take the blood sample, extract it, isolate out just the oil part, measure it. Then came the 80s. If you look at your blood report now, you'll see something called HDL, LDL, VLDL ratio. Those are all naturally water-soluble forms of cholesterol. That's why they can pass through the blood. Chlorophyll, which is the green pigment of plants, there's a chlorophyll binding protein that can instantly make it water-soluble. So there are lots of examples in nature, and these have been known for at least 3 decades, that can make an oil stably, naturally water-soluble. And that's the part we have focused on, that plants actually do have naturally water-soluble molecules. And from a basic science perspective, that's what you'd predict. Otherwise, the molecule will never be able to move around because the living fluid is water-based. So the molecule has to be in a water-soluble form.
We're going to move on to a question over here now.
Okay. Hi. So with these biosynthesized molecules, like you're describing, so you're adding a compound to a known molecule, THC, CBD, changing it. How much study have you done on how that is digested? Like, does it go through the endoplasmic reticula? Does it digest like a lipid? Where does it go into the bloodstream? And how does that fit into the regulatory framework that exists today? And is it known to be safe? Have you done studies on it?
So our glycosylated cannabinoids that I can talk to you more about, that also occurs naturally in the plant. Glycosylated cannabinoids occur naturally in the plant. And our process of glycosylating them out of the plant is also a natural process. And then all the fluids, the digestive fluids, starting with saliva, are loaded with glycosidases. So 18% of our saliva is glycosidases. So in the oral phase alone, it reverts back to the parent compound. And the toxicology for CBD and THC is known. So, you can show that it doesn't go into the bloodstream in the glycosylated form. That helps you.
I think it depends on your perspective. If you're truly looking for a traditional safety study, it's going to be very difficult to do because the proper analytical technology doesn't exist. In the pharmaceutical world, before you ask those kinds of questions, there is a series of studies that are done called ADME, absorption, distribution, metabolism, excretion. Unless you know all those features of a chemical molecule that is administered through different routes and different doses, oral versus injection, right? You have to know all of those before you can truly study safety. Because if in one mode it just gets completely distributed into liver, it'll have one safety or lack of safety related issues, as opposed to it just uniformly circulates in blood, some goes into brain, the rest gets excreted. It'll have a completely different safety profile. And that's one of the things that goes back to the nanotechnology. The FDA got concerned back in the 80s that you're actually changing what's called the pharmacokinetic and pharmacodynamic property of the molecule. So in more ways than one, it's called an NCE, or a new chemical entity, by the FDA definition, which means now you got to start all over again because you simply don't know what this molecule, this new molecule, is going to do.
An emulsion doesn't create a new molecule. If I understand it correctly, the biosynthesis does create a new molecule, but an emulsion doesn't.
Biosynthesis can, but it doesn't have to. For example, through complete what's called metabolic pathway cloning, you can insert all 17 genes that are involved in converting the precursor into THC or CBD. You can put them all in yeast, right? And make the yeast use its natural nutrients, be it galactose, sugar, and all of that, take it all the way out to the cannabinoids. When excreted, most likely it'll be produced in the acid form, which is water soluble, way more water soluble than the decarboxylated delta 9 or delta 8 form, right? So that can be done as a molecule that is exactly the same as what the plant had made. And that's, again, foundation of biotech. Insulin, you can go collect pancreas from slaughterhouses, purify insulin, or you can put simply the human genetic information that encodes insulin in a bacteria, make it produce the protein. The two proteins are identical, all right? But again, there are also ways, as Jennifer pointed out, to modify these molecules mimicking what Mother Nature would do. Plant would do it slightly differently than humans versus cats versus rats. And each one, unfortunately, may have different consequence.
Hi, this is just following on with the same kind of thought, I think. But if you can comment on the bioavailability of the molecule, so like CBD, for example, if it's using nano-emulsification or glycosylation, is this something that would improve the bioavailability to the receptors, or just if you can comment on that?
Again, the term bioavailability, as it's used in this industry, is total non-science. They indicate bioavailability based on you ingest something, how much of it is appearing in blood, what form, and so on and so forth. Again, as I went back, if you have not done the ADME study on the molecule, just knowing how much gets into blood is absolutely meaningless. And this has happened with the diet supplement industry. People make different forms of calcium. Mine is more bioavailable than his. That simply means more of it shows up in blood. What is the functional role of calcium? Primary role is strengthening bone, mineralizing bone. So if it's not going to the bone, what difference does it make, right? So bioavailability as done simply coming from 3 decades in biotech and pharma industry is absolutely meaningless.
So you don't think there's any bioactivity relationship between these?
Minimal, absolutely minimal, because what is, let's say, THC supposed to do?
Well, CBD used to be.
Same thing. CBD can work on multiple different cells. So if it specifically goes into one organ, it'll have one activity profile that might be totally different from it going somewhere else.
I guess what my question is these technologies, how do they increase the availability in the blood if it's oral?
So the glycosylated CBD, for example, because it begins absorption in the oral cavity and continues through your digestive system, it kind of digests it for you in a way. So the experience is more like inhaling the plant material than it doesn't have to go through the whole enzymatic process in the liver. So there's a very predictable and shorter onset time, and there's a very predictable and offset time. So you can do actual dosing studies. You can do real pharmacological dosing studies, and with a hydrophobic compound, you have to base your dosing on the individual, based on their individual metabolism, their enzymatic levels in their liver. With a truly water-soluble cannabinoid, you can do dosing that's generalized to that age group, that male or female. You can much more general dosing, and you can actually do the pharmacological dosing studies. It's impossible with a hydrophobic compound. So if that answers more of your question, the water-soluble ones are more bioavailable. But I agree with Dr. Sen, that's not the best term, really.
Let me address that too. So if we just stay with the definition of what bioavailability means, it's the ratio of the compound that arrived in the bloodstream. There's a pharmacokinetics and pharmacodynamics. These are two separate things. Bioavailability is just responsible for the arrival at the blood. So if your question is on that, then there's some pretty straightforward answers. The bioavailability goes up depending on how good your formulation is for nanoemulsions. I'm not sure for the water-soluble compounds, roughly by a factor of 5 to 7. And that is based, unfortunately not yet, although we're in the middle of a study like this right now, it's based on other hydrophobic actives being delivered that way compared with when they're delivered just as oil capsules. The bioavailability increase seems to belong more to the vehicle, to the method of delivery, than to the drug itself. So nanoemulsification and the bioavailability studies have been done on many drugs, not so much on cannabis yet, although we hope to have some data soon, probably by the next time we're here, if we're lucky. There is also another thing to consider, is direct blood absorption and then liver. So yes, first pass effect versus lymphatic absorption and then going around the liver, and therefore no first pass effect. So things get destroyed in the liver when they arrive there first. With THC, it's more complicated because you actually make hydroxy-11, which has its own action, and it's more active in some ways. So it's really hard to really say what you're experiencing. This is beyond bioavailability now, right? This is the effects already. Now, CBD simply gets destroyed. One thing that is perhaps advantageous about delivering things in their hydrophobic form, but in the water-compatible form, so not as truly soluble compounds, is that you can then direct them into the lymph. And that is very advantageous, especially for CBD, because you can, if you deliver them as a nanoemulsion, that mimics the mixed micelle formation. I'll have a lot more in my talk later on today. They get absorbed into the enterocyte cells where if it's co-administered with fats, basically on a full stomach, it will get incorporated into a chylomicron, and that will take it into the lymph, going around everything. In that case, the bioavailability increases even further. And also another consideration is the onset time. So when it goes straight into the blood, it's a little faster, although it gets destroyed. So the onset is there, but the intensity is reduced. When it goes into the lymph, it'll have to go around the liver, and then it gets into the blood above your heart. And that takes a little longer, so there's like an additional maybe 5-10 minutes delay. Although in our studies now, for some reason, we're not seeing a lot of that. We're seeing maybe an additional 2-3 minutes delay. In 10 minutes, it's pretty clearly felt, which means it's not only bioavailable. It didn't only arrive in the blood, it actually has the effect, so subjects reported. But it's a little faster when it goes straight into the blood.
We have one last question, but then our panel will also be on the floor for a little bit, so last question for this session.
Kind of on the onset and offset, how consistent have your studies been on those, on the 3 different technologies? And then also, is it mandatory that the mixture be put in like a light-proof container when packaging?
Sorry, the difference between the onset times when you compare these 2 approaches, is that what the question was?
Yeah, like how, what are your current onset and offset times, and are they consistent throughout all your testings that you've done so far?
Yeah, for nano emulsions, it's pretty rock solid. It's about 10 minutes. And this is for the effects. We're talking about THC. We haven't done the proper study yet. Again, we're in the middle of one. So I suspect that it arrives in the blood before that, because then it has to also get to the brain, and you have to become aware of the effect. So about 10 minutes. On a full stomach, maybe 15. Consistent, yes, except on a full stomach, because that depends on what else you ate. And that would be true for anything, simply because of gastric emptying. So things hang out in your stomach and wait their turn to empty into where they can get absorbed. You know this from alcohol, and alcohol works the same way. So for nanoemulsions, and most likely for truly water-soluble, maybe even more so for truly water-soluble compounds, you basically approach what alcohol does. So the logic becomes more familiar, more predictable, and basically everything works progressively more that way.
Yes, for the water-soluble, we're also in the middle of studies on that. To give you a precise answer, we're doing some clinical research in Israel with the water-soluble THC at the moment. And those aren't complete yet. But I can tell you it's about a 5 to 10 minute onset time. And
is the offset consistent as well, or is it
3 hours.
3 hours usual?
for us it's about an hour and a half. And the benefit of that is you don't have the people eating or drinking too much before they realize it. So you can dose yourself in a more predictable way. It's
like vaping through your small intestine.
Exactly, yeah.
So our technology, which is recovering the naturally water-soluble forms from the plant itself without changing anything, we have now done 21 different varieties, about 16 cannabis strains, 5 hemp CBD, quote-unquote, CBD strains. In every single case, first of all, we feel the effect in less than 5 minutes, bar none. There is no difference at all. And it's always gone by 90 minutes, most often 60. Now, these are obviously, as Alexey pointed out, the cognitive effect. Whether or not the blood level is low enough so they're not feeling anything, that details, it is going to come in the future, not available now. So from my perspective, for THC especially, offset predictability is extremely important, because unlike beneficial oils, you do not want the THC oil form to hang around in your system forever, which is exactly what happens with the edibles, the smoking, vaping, and all of that, right? So there are some unique benefits in naturally water-soluble material. There's a reason why Mother Nature created it that way. In our body, she created HDL and LDL, so there are definite reasons for it. And that's why trying to stay with the natural matrix, not disturb it until you know what it does, how you want to change it. It's just a more reasonable scientific approach.
Thank you so much to our panel. Let's give a round of applause for everyone.